Very good morning to all my dear students. In the last class, we started the fifth lesson molecular genetics, and in the last class, we studied about some important 5-mark questions. The first question we studied is the properties of genes, then the nitrogenous base, okay, the chemistry of nucleic acid, nucleotide, nucleoside, like that many 5-mark questions and some 2-mark questions, 3-mark questions also we have studied. And some students will send the answer to me through WhatsApp. I really appreciate you. Then today we are going to study about some topics. Okay, so listen very carefully. Every day study the lessons, study all the lessons. Today we are going to study about the first topic is RNA value. See, the last class I told you the two types of genetic materials are there. One is called DNA and the second one is called RNA. And in some organisms, DNA is acting as a genetic material, and in some organisms, the RNA is acting as a genetic materials. Understand? So today we are going to study about RNA bio. Say, inside the cell, the RNA molecules playing more function, more varieties of functions inside our cell than the DNA molecules. Okay. So the RNA is playing very important functions and the different varieties of functions inside the cell than the DNA molecules. So the number of RNA molecules is 10 times higher or more than the DNA molecules. The number of the RNA molecule is 10 times more than the number of DNA molecules. Understood this point? Then, say 3 scientists, Frankel, Conrad and Singer. These two scientists only, by doing one experiment, they clearly said in some organism, the RNA molecules are acting as a genetic material. Example, tobacco mosaic virus. So in tobacco mosaic virus, the RNA is the major genetic material. Okay, it was said by these 3 scientists. Then one scientist. His name is Walter Gilbert. He only first introduced this term RNA back in the year of 1986. So it's one of a very important 1-mark question. The term RNA world was first introduced by Walter Gilbert in the year of 1986. Okay, and he said this is a first genetic material. RNA is the first genetic material in the earth. Okay, so in the olden days, maximum the living organism, he said the living organism, this RNA is the first genetic material. In the olden days, the living organisms, inside of the living organism's body, the nucleic material is RNAs. Okay, then next 3 scientists, Leslie Worgel, then Francis Crick, then Carl Woese. So these 3 people or these 3 scientists togetherly said the RNA is the first stage in evolution. First stage in the evolutionary process. Here, first stage means the stage in which the activities or the metabolic activities of the organism's body is catalyzed for their survival and for their replication. Understand? So, in some stages, some metabolic activities are speed up or catalyzed by some substances. And these activities are very, very important for the survival and also for the replications of the living organisms. Okay, so these 3 people togetherly said this is the first stage. RNA is the first stage in the evolution. Then, next, these RNA molecules act as both genetic material and also it acts as catalyst. Catalyst means already I told you to speed. Some substances are used to speed up the reactions inside the body cells. Inside the body cells of some living organisms. So here this RNA is acting as a catalyst. For example, in our body, ribosome. This is one of the example for the catalyst. Then next, these RNA molecule can act as a gene regulators. Here gene regulators means some RNA molecules bind with the DNA molecules. Some RNA molecules are bind with the DNA molecules and it control or affect the genes expression. So once again repeat this point, some RNA molecules are binding with the DNA molecules and affect the genes expressions or affecting the genes functions. So some RNA molecules are acting as a gene regulators. Then the last point about this RNA body is two scientists, Andrew Farr and Karl Mello. These two scientists says this RNA is one of the very important active ingredients in the chemistry of a life or in the chemistry of the living organism. Understand? So these are the some of the points now we have completed about this RNA word. Hope you understood this RNA word. So this RNA word is one of the very important 5-mark question. That's all. Once again repeat. See, Frankel, Cantor and Singer, these three scientists only first discovered that this RNA is the genetic material in some organisms like tobacco mosaic virus. And inside the body cells, the RNA playing different varieties of roles. So the number of RNA is 10 times more than the DNA. First point. Then the second point, Walter Wilbur first introduced this term RNA word in the year of 1986. And he said this RNA word is the first genetic material in the earth. Then next, Leslie Swargel, Francis Crick and Carl Woese, these three people togetherly said this RNA word is the first stage in evolution. Here first stage means the stage in which the process, the metabolic activities are speed up or catalyzed by some substance. Then next point, this RNA acts as both genetic material and also it can act as a catalyst. One of the example for this catalyst is ribosome. Then the RNA molecule can act as a gene regulators. That means some RNA molecule bind with the DNA and affect the gene's expression. Then the last point, Andrew Farr and Karl Mello, these two people togetherly said this RNA is one of the very important and very active ingredients in the chemistry of the life. Okay, my dear friends. Okay, I hope you understood this question. Then the next question is, okay, the next question is properties of genetic materials. Properties of genetic materials. Okay, see, genetic materials meaning nucleic acid. Okay, the material which is transferred from one generation to next generation, or the materials which is transferred used to control the activities of the cells. Okay, or activities of the entire organism is called genetic materials. Okay, so now we are going to study about the properties of genetic materials. See, in the last class I told you two scientists, Hershey and Charles. These two people said the DNA is the genetic material in maximum living organisms. Okay, because some people during that two people's period, some scientists believed that the protein molecules are acting as a genetic material. Okay, but these two peoples togetherly involved in, they have done one experiment to prove that the DNA is the genetic material. So that experiment is called this experiment. Okay, so this experiment clearly they said DNA is the genetic material. Okay, so some scientists believe that the protein molecules are the genetic material and some scientists believe that the DNA is the genetic material. Okay, so now we are going to study about the properties of genetic material. Okay, the first property is self-replication. The first property is called self-replication. Listen very carefully. See, self-replication meaning, see, according to the base pair rules and regulation. Base pair means, already I told you in the last class, see, adenine should combine or pairing with the thymine. Guanine should combine or pairing with the cytosine. Okay, so this is the base pairing rule. So according to this base pairing rules, the both DNA and RNA, the genetic material should have the capacity to replicate. Okay, so should have the capacity to replicate. So that's all about this first property is self-replication. So once again, I repeat this point. So based on the base pairing rules and regulations, both genetic material, both DNA and RNA should have the capacity to self-replicate. Understand you? The next property is stability. Next property is stability. See, stability meaning, see, the genetic materials inside the cell of a living organism The genetic material should not change their structure or their chemical nature under any situations. See, inside the body cells, the genetic material, whether it is DNA or RNA. Okay, so the genetic material should not change their structure or should not change their chemical component under any stage, any conditions. Some life, for example, some life organisms have some stages. Okay, so in all the stages, the DNA or the RNA, the genetic material should be in a constant stability. It should not change. Or in some the genetic materials should not change during the age. Okay, childhood stage, adult stage, okay, old age stage. So during these stages also genetic material should not be changed under any situation. Okay, so this condition is called stability. Okay, then one experiment. Okay, this experiment was done by Griffith, scientist Griffith. So he used one experiment with the help of some bacteria. Okay, some bacterial species are heated in high temperature. Okay, so when he applies some heat, overheat to this bacterial species, some important genetic materials is not changed inside the bacterial cells. Understand? Then in our body cells, okay, double-stranded DNA is there. See this, when we separate this double-stranded DNA by using overheat or over temperature, it will separate. Okay, but when the condition is favorable, automatically the separated DNA is joining again. Understand? So like that, the nucleic materials or the genetic materials should not change their nature under any conditions. Understand? Then in RNA, already in the last class I told you, in RNA additional one OH group also there in the second position. Okay, one additional OH group also present in the second position. So because of the presence of this additional OH group, the RNA is less stable and easily degradable, but the RNA is very reactive, more reactive than the DNA. Once I repeat this point, say in RNA one more additional OH group, that means two OH groups are present. So because of the presence of this additional OH group, it is more active, more reactive and less stable, and it can easily degradable. Understand? But the DNA is more stable but at the same time less reactive. RNA is less stable and more reactive. The DNA is more stable but less reactive. I hope you understood. And in the last class I told you, in our DNA, instead of uracil, thymine is present. Okay, uracil is present in RNA, thymine is present in the DNA. So the presence of this thymine also giving additional stability to the DNA molecule than the RNA molecule. So the second properties of the genetic material is stability. Here, the stability of the genetic materials, whether it is DNA or RNA, should not change under any situation. Should not change in the stages of life cycle or should not change by age. Understand? So this is the first point I said. The second point, one experiment has done by scientist Griffith. Okay, he has done one experiment, and this experiment he given some heat, more heat to the bacterial cells. Instead of this overheat, the bacterial cells in some properties, some important properties of the bacterial cells are not changed. Okay, then the next point, in our body cell double-stranded DNA is there. When we separate this double-stranded DNA by using overheat, when the conditions are favorable, again these two separated strands are joining togetherly. Understand? Then the next point I said about the stability is in RNA one more additional OH group is present. So because of this OH group, the RNAs are more active, more reactive. Understand? And it can easily degrade. Understand you? Then the DNA molecules are more active, more stable and less reactive. And one of the additional reason for the more stability of DNA molecules is presence of thymine. Because of the presence of thymine, the DNA molecules are more stability than the RNA molecule. So I hope you understood these 2 points. Then the third property is information storage. The third property is information storage. The genetic material should have express their characters in the form of Mendelian characters. Mendelian characters, in the last class I told you, a characters controlled by a single gene is called Mendelian characters. So the genetic materials, whether it is DNA or RNA, should have the capacity to express their function in the form of Mendelian characters. So this is the first point about this information storage. Then the DNA molecules, listen very carefully, DNA molecules cannot synthesize any protein molecules directly. So to produce the protein molecules, the DNA is depending RNA molecules to produce protein molecules. But RNAs can directly synthesize protein molecules. So the second point about this information storage is DNA molecules are always depending the RNA molecule for protein synthesis, and the RNA molecules directly they can synthesize the protein molecules. So now I said two important points about this information storage. Understand a few? Then the RNAs can transfer the genetic information, but the DNA can store more genetic information. So the third point about this information storage is RNA can transfer more genetic information, but the DNA can store more genetic information. Okay, so now I said three important points about this information storage. Hope you understood. Then next, the last property is variation through mutation. Variation through mutation. Okay, variation through mutation. See, listen very carefully. Both DNA and RNA should have the capacity to involve in mutation. Okay, and already I told you the RNA molecules are less stable than the DNA molecules. So the RNA molecules can mutate very fastly than the DNA molecules. Only 2 points about this. Variation through mutation. Both genetic materials should have the capacity to involve in the mutation, and maximum the RNA molecules are mutating very fastly, involving in mutation very fastly, because RNA molecules are less stable. Understand, my dear students? Okay, see, now we have completed one important 5-mark question, properties of genetic materials. There are 5 properties are there, 4 properties are there. The first property is self-replication. Okay, self-replication. The second property is called stability. Third property is called information storage, and the last property is called variation through mutation. I hope you all understood this point. Then the next question is packaging of DNA. Packaging of DNA. See, listen very carefully. This is little big question and the points also very difficult to understand. So listen very carefully. Packaging of DNA. See, base pairs you all know. Okay, adenine, thymine, guanine, cytosine. Okay, this is an example for base pair. See, a distance between two base pair is 0.34 nanometer. Listen very carefully. The distance between two nearest or two adjacent base pair is 0.34 nanometer. Okay. Then the total length of our DNA helix is 2.2 micron. 2.2 micron. Listen very carefully. So the distance between two adjacent base pair is 0.34 nanometer and the total length is 2.2. See, to find out the total length of the DNA molecule, we have to multiply the total number of base pairs with the distance between two adjacent base pairs. So, total number of base pair is multiplied with the distance between two adjacent base pairs. We will get the total length of nucleic acids, total length of DNA. Understand a few? Then, for example, in E. coli, okay, listen very carefully. In E. coli, the total length of the DNA is 1.36. Okay, 1.36 nanometer. Total length is 1.36 nanometer. Then the number of base pair present in the Escherichia coli bacteria is 4 into 10 raised to 6 microns. Understand? So now I said 2 points here. Total, the distance between two adjacent base pair is 0.34 nanometer and the total length of the DNA is 2.2 micron. Okay. Then to find out the total length, the total number of base pair is multiplied with the distance between two adjacent base pair. And this is the total number of the total length of the DNA of bacteria, and this is the total number of base pair present in the bacterial cells. Hope you understood this point. Then the next point is maximum the genes, the chromosomes only carrying the genes. Okay, in eukaryotic organisms, maximum organisms body cell, the chromosomes are there. Okay, so the chromosomes only carrying the genes and the genes only transferring the characters from one generation to next generation. From one generation to next generation. Then in plants and animals are the example for the eukaryotes. Okay, so in plants and animals, more number of DNAs are present. Okay, more number of DNAs are present, and all these DNAs are held or kept inside the nucleus. But in prokaryotic organisms like bacteria, there is no nucleus, and the DNA number of DNA also very less, and the DNAs are not scattered inside the cell. The DNAs are particularly present or kept inside a protein. Name of this protein is called nuclei. Name of the protein is called nuclei. Listen this point very carefully. In plant cells and animal cells, the number of DNAs are more, and all these DNAs are kept inside the nucleus. But in bacterial cells, there is no nucleus, and these nucleic materials or the DNAs are not scattered inside the cell. The nucleic materials or the DNAs are kept inside a special type of protein. Name of this protein is called nuclei. I hope you understood this point. Then the next point about this packaging of DNA is one important 2-mark question. Genophore. See, genophore means in prokaryotes, maximum the DNA molecules are circular in shape and lack of chromatin. That means there is no chromatins are present inside the nucleic material or DNA. So the DNA in prokaryotic organisms, the DNAs are circular in shape and lack of chromatin. Chromatin is absent. So this condition is called genophore. But in eukaryotic organisms, the chromatins are made up of many small subunits, many repeating subunits. Name of this repeating subunit is called nucleosome. Name of this repeating subunit is called nucleosome. Understand? And this nucleosome is positively charged molecules. Nucleosome is a positively charged. So just imagine it's a nucleosome, it's a positively charged molecules. And the DNA molecule is covered or warped the outer surface of the nucleosome. Okay, and maximum the DNA molecules covering this nucleosome 2 times. Okay, 2 times they are covering or warping the outer surface of the nucleosome. So I hope you understood this point. Then, word scientist, his name is called Kohlberg. See, he said the nucleosomes, okay, this is the structure of nucleosomes. So, the nucleosomes are made up of 2 molecules. Nucleosomes are made up of 2 molecules. Each molecule is made up of 4 histone proteins. 4 protein molecules. Name of this protein molecule is called histone protein. Okay, so a single nucleosome is made up of 8 protein molecules, 8 histone protein molecules. Okay, so all these 8 histone protein molecules are called histone atoms. Is called histone. I hope you understood this point. See, Kohlberg is a scientist. He said that the nucleosome is made up of 2 molecules. Each molecule is made up of 4 important histone proteins. So a single nucleosome is made up of 8 protein molecules, 8 histone protein molecules. It is called histone atom. Yes. Okay, it is called histone atom. Yeah. Hope you understood this point. Then the next point is, see, a single nucleosome consists of 200 base pair of DNA pairs. A single nucleosome is made up of a single nucleosome consists of 200 base pairs. Understand? Then the adjacent nucleosomes or the nearest nucleosomes are connected or linked by a DNA. It is called linking DNA or H1 DNA. Or is called linking DNA or is called H1 DNA. Hope you understood this point. See, two adjacent nucleosomes are connected or linked by a DNA. It is called linking DNA or is called H1 DNA. Understand a few? Then the next point is, see, in some chromatin, this H1 DNA or the linking DNA is absent. Okay, some chromatin, the linking or the H1 DNA is absent. So these chromatins appear like a beads on a string. That means, just imagine, say, nucleosome. The DNA molecules are entering inside the nucleosome and leaving, coming outside. Then entering inside the nucleosome, then coming outside. But here not like that. Okay, the nucleosome which are having, the chromatin which are having the linking chromosomes, the linking DNA is moving outer surface of the nucleosome. But here the DNA is entering inside the nucleosome and coming outside. So it is look like a beads on a string. I hope you are understood this point. Understand? Okay. Listen the next point very carefully. See, now I told you in some chromatin this linking DNA or H1 DNA is absent. So inside this, in this type of chromatins, the nucleus and the DNAs are entering inside and coming outside. Okay, entering and leaving outside. Then next point. See, maximum the chromatin fibers diameter is vary from 200 to 300 diameters. That means in some chromatin fiber the diameter is 200 nanometer, and in some chromatin fiber the diameter is 300 diameter. So the diameter of the chromatin fiber is vary from 200 to 300 diameter. Understand? Then 30 nanometers. Okay, 30 nanometers of chromatin fibers are arising from the foldings. And all these 30 nanometers, okay, 30 nanometers are togetherly making a chain-like structure. This chain-like structure is called solenoid. This chain-like structure is called solenoids. So listen very carefully. The diameter of the chromatin fiber is differ or vary from 200 to 300 diameters. Okay. That means in some chromatin fiber the diameter is 200 nanometer. In some chromatin fiber 300 nanometers are there. So maximum 30 nanometer chromatin fiber is arising from the folding of the nucleus. Understand? And they are togetherly forming a chain-like structure. It is called solenoid. It is called a solenoid. Understand a few? Then the DNA molecules are packed with 40 foldings. There are 40 foldings are present inside the DNA molecule, and one more additional set of proteins also needed for the packing of this DNA. Name of that proteins are called NHC protein, non-histone chromosomal protein. Listen very carefully. There are 40 foldings are present. Okay, so to pack all these DNA molecule, one more set of protein is very important. That protein name is called NHC. Okay, non-histone chromatin protein. Sorry, non-histone chromosome protein. Understand a few? Then the last point about this packaging of DNA is inside the nucleus. In some region of the chromatin is loosely packed. Some region of the chromatin is loosely packed, so that region is called euchromatin. This region is called euchromatin, and this region is transcriptionally very active. This region is transcriptionally very active. Then in some areas or some regions of the chromatin is tightly packed or tightly arranged, and this region is called heterochromatin, and this region is transcriptionally inactive, less active. So let's repeat the last point about this question. Inside the nucleus, some region of the chromatin is loosely packed or loosely arranged. This region is called euchromatin, and this euchromatin, and this euchromatin region is very active. Transcriptionally, it is very active. But some region, the chromatins are tightly arranged or tightly packed. This region is called heterochromatin, and this heterochromatin region is inactive in condition. Transcriptionally inactive in condition. Understand my difference? So now we have completed one important 5-mark question and many 2-mark question. This length, size, number of base pairs, then this keystone atomier, then one 2-mark question. I said genophore meaning in some prokaryotic organism, the nucleus, that means DNA is circular in shape and it has no chromatin. So this condition is called what? This condition is called genophore. Okay. Then the keystone atomier. Keystone atomier meaning maximum a single nucleosome is made up of 2 molecules. Each molecule has 4 keystone proteins. So totally 8 protein molecules or 8 keystone protein molecules are together forming a keystone atomier. Understand that you? Then this positive charge, negative charge maximum. These nucleosomes are positive in charge. Then the DNA molecules are negative charge. Okay, like that many 1-mark questions also there. So I hope today you understood all these 3 question answers clearly. So today is self-study and send the answer to me. Okay, we'll see you in the next class. Thank you.